flower? Pretty, isn't it? It's Developmental. A little interesting thing about it is that it has a reduced perianth, but instead the stamens are petaloid, so they take on petal characteristics. They're large, purple, and showy to attract pollinators. It's amazing how diverse flowers can be. I didn't formally introduce myself. My name is Jesus. I'm an undergraduate here at the University of Washington in Seattle, and I study evolution of flower development in Verónica Stilio's lab. Come on, I can tell you a little bit more about it. We can get out of this noisy place. As I mentioned in the greenhouse, I'm interested in understanding the evolution of flower development. So what that means is, I'm trying to understand how the genes that produce the different organs within a flower have evolved throughout time. So before I go more into that, first let me do an introduction into the anatomy of a flower. So in the first row, we have the sepals, which are the leaf-like structures that encase the flower when it's in a bud. In the second row, we have the petals, which are used to attract pollinators. In the third row, we have the stamens, which are the male reproductive organs of a flower, and those produce the pollen. And in the middle, we have, or in the fourth row, we have the carpels, which are the female reproductive organs of a flower, and they produce the seed. So the ABC model of floral development, which was established in Arabidopsis and in Snapdragon, predicts which groups of genes are needed to produce which floral organs. So what that means is, if we have A-class expression in the first row, we'll have sepals. If we have a combination of B-class expression and A-class expression in the second row, we'll have petals. And if we have a combination of B-class expression and C-class expression, we'll have stamens. And finally, C-class expression alone will give you carpels. So I'm interested in understanding how these B-class genes have evolved throughout time in a different plant, not any of the current model organisms. So let me go show you. This is the Untitled, commonly known as rue anemone. It belongs to the Ranunculales, a lineage sister to all other eudicots. Unlike Arabidopsis, it doesn't have any petals, but instead the sepals serve a petal function. So these white organs are the sepals, and they look like petals. So I'm interested in characterizing the B-class genes within the Untitled. And if B-class genes specify petals in Arabidopsis, could they perhaps be specifying petaloid sepals in the Untitled? In order to answer that question, I use a lot of different molecular biology techniques. I can go show you them. Come on. Most of the time, I'm here at the bench doing a lot of molecular biology. And that includes PCR, molecular cloning, and most recently, a lot of yeast-2 hybrid. In order to make sure that my experiments are a success, I gotta make sure that my lab bench is clean, I'm using sterile technique, and most importantly, that I'm wearing my lab coat. So now, let me go show you where I analyze my experiments. This is where I analyze my DNA sequence. After a long day at the bench, it's always nice to kick back and analyze some data. Look at all those mutations! Now, let me show you the best part of my job. Just kidding. This isn't my favorite part of the job. But if you make a mess, you gotta clean it up. I think I'll wait for tomorrow to wash these. Thanks for stopping by. I hope you learned a little bit. I'll catch you next time.
Whose dishes are these?